be the issue. It might have just been that I didn't press the button all the way.
Okay, guys. So chapter 12, we're talking about eukaryotes. You guys tell me, what are some eukaryotes?
Plants, algae, fungus,
Animals,
animals.
people.
You got protozoa,
Parasites.
you got plants, you got algae, you got fungi, parasites, animals, people. So these are made up of eukaryotic cells, cells that have a nucleus and membrane-bound organelles like mitochondria, Golgi apparatus, all that good stuff. One thing to know is the difference between mitosis and meiosis. So you guys tell me, what's the difference between mitosis and meiosis?
This is individual cell reproduction.
Mm-hmm.
That ends up with a diploid, 2 sets of chromosomes.
Mm-hmm.
Meiosis, it's 2 rounds of division.
Uh-huh.
Ends up with 4 daughter cells and 1 set of chromosomes.
Yes, so what she said. Mitosis is individual cell reproduction. So think of that, a skin cell making another skin cell. You start off with a diploid mother cell. That means a mother cell that has 2 sets of chromosomes. You go through the 4 rounds of division, prophase, metaphase, anaphase, telophase, cytokinesis, and you end up with 2 daughter cells that are also diploid. So the 2 daughter cells from mitosis are identical to the original mother cell. Meiosis is only used for sexual reproduction in eukaryotes. And it's used specifically to make the gametes, like sperm and eggs. So typically it only occurs in the gonads, the testes and ovaries. And with meiosis, think of it like mitosis times 2. There's actually a second round of division. There's literally a prophase part 2, and a metaphase part 2, and an anaphase part 2, etc. So you end up with 4 daughter cells, but they are haploid. What's that mean? They only have 1 set of chromosomes. So they are not genetically identical to the original mother cell. So that's the big difference. So please know the difference between mitosis and meiosis. Now, there's also schizogony, which you see with some protozoa and some fungi. This one's where they have multiple series of mitosis, but they don't go through cytokinesis till the end. Because remember, mitosis, strictly speaking, just means division of the nucleus. Most of the time, cytokinesis happens at the same time. But with schizogony, which you see in some protozoa and some fungi, they divide their nuclei up multiple times and then decide to do cytokinesis at the very end and release a whole bunch of daughter cells at the very end. But that's more the exception than the rule. Okay, now talking about specific eukaryotes, what are protozoa? Well, these are single-celled eukaryotic organisms. They do not have a cell wall, and they live in water. They live in moisture, and they are very motile. They move around either by flagella, by cilia, or by the pseudopod method. And if they move around by the pseudopod method, they're also known as amoebas. So amoebas are just protozoa that use pseudopods. Protozoa that use flagella or cilia are not amoebas. And once again, you always find them in moisture, either ponds, streams, lakes, the oceans, little creeks, rivers, etc. Even in moist soil. Very few are pathogenic, but the ones that are can be really problematic. They have 2 different life cycles. They have a motile feeding stage where they're swimming around trying to eat stuff. That's known as the trophozoite stage. And they have a resting stage they call cystage, where they kind of curl up in a hard little ball and rest. And this is your typical protozoa. It's a single-celled eukaryote. You can see it has a big old nucleus. It's got what we call a vacuole. It's literally like a water pump. And then if you look closely on the plasma membrane, you can see these little tiny hairs, which are cilia, and that's basically what kind of moves it through the water. And they basically eat bacteria, other protozoa, decaying matter through the process of phagocytosis. Specific protozoa you should know, one would be Trichomonas. And this is one that unfortunately can cause an STI. Trichomonas is a protozoa that loves to live in kind of moist, warm, slightly acidic environments. And the human urethra is warm and moist and slightly acidic because urine has a pH of about 6.8. So this one is the STI that can be transmitted from partner to partner. What medication do you use to treat trichomonas? Metronidazole. So remember, that's okay. It used to be called Flagyl. And I'll be honest, everyone and their brother or sister still calls it Flagyl. It's a lot easier to say Flagyl than metronidazole. But metronidazole, which also goes by the name Flagyl, is technically an antifungal agent, but it's really good at killing protozoa. We don't know why, but factually speaking, it kills protozoa as good, if not better, than fungus. So I actually say it's actually antifungal, anti-protozoa, because metronidazole is pretty much the gold standard what you give somebody if they have a protozoa infection. So we'll talk more about that at a different time. Giardia is another protozoa you should know. What's significant about Giardia? That's the one that's in water, and if you don't boil the water, Giardia will infect your intestines and give you severe dysentery. It's found in most freshwater lakes, ponds, streams. That's why if you go camping or something, and you should boil the water at least 15 minutes or put an iodine tablet in it. Otherwise, you're likely going to get a Giardia infection, which is severe dysentery and is no fun. Another protozoa I think is significant is Plasmodium. You guys tell me, what's Plasmodium do?
Malaria.
That causes malaria. Now, Plasmodium is a protozoa that's found in the blood of monkeys. It doesn't make the monkey sick. But if a mosquito bites the monkey and gets that protozoa in its saliva, and then it turns around and bites a human, the human will get very sick. Because Plasmodium likes to set up shop in the liver and give you hepatitis, which will give you jaundice. It also likes to eat human red blood cells. So it's not basically a disease you want. So that's Plasmodium, which causes malaria. And then finally, the last one I think is important for you to know is Naegleria fowleri. That's the full name. So what does Naegleria fowleri do?
Brain-eating amoeba.
That is the brain-eating amoeba. And how do you get it?
Through the nose.
Usually water going up the nose. So Naegleria fowleri is an amoeba that loves warm water. And it's very prevalent in the south, especially in the summer months, in lakes and ponds. And if it goes up your nose, it's going to break through that ethmoid bone and start eating your brain, literally. And it unfortunately has a very high fatality rate because it's usually not diagnosed in time. Its symptoms are pretty much like any other type of encephalitis or brain infection. So doctors usually are trying to rule out, like West Nile and Zika virus and 20 other things. And by the time they get to thinking about amoebas, it may be too late. So always keep in mind if your patient was swimming recently, it's the summer, Naegleria fowleri is always a possibility.
About how long would it normally take from the swimming incident too?
Usually it's about 3 days, and the patient starts to get delirious and starts to get disoriented, and usually have a seizure, etc. And if they're treated immediately with like antifungal agents, like IV metronidazole or amphotericin B, they could survive. The problem is they may not get diagnosed for another 2 days, and by then there could be significant brain damage. Now, there are cases where patients have survived this, but it's rare. But it's usually because they were diagnosed early. So, all right, so those are your protozoa to know. Fungi are another type of eukaryote. Remember, fungi are not plants. They're actually more closely related to animals and people than plants. They do have cell walls. They're made of a hard protein called chitin. They don't perform photosynthesis. And they are basically nature's recyclers. That's pretty much the goal of fungi. They help decompose dead organisms, both plant and animal, and try to recycle their nutrients for the environment. So they can help plants absorb water and minerals. Some of them are useful. Obviously, we use some of them to make antibiotics, and some of them we can eat. But there's a few that are pathogenic. And they can either be molds or yeast. What's the difference?
Yeast buds and
Yeah.
Mold is multicellular.
Yes. So you're both right. Molds are multicellular. That means they are multi-cell organisms. They tend to have these long filaments called hyphae, and that's kind of what gives mold its kind of fuzzy appearance. By comparison, yeast are single-celled fungi. They're globular, and they usually reproduce by budding. So this would be a mold. It's got these really long filaments called hyphae. This is what gives mold its kind of fuzzy appearance. And at the end of these hyphae, it have spores, and those spores, what it releases into the environment and helps it spread. By comparison, a yeast is single-cellular fungi, and they reproduce by budding. Most of the fungi I want you to know is actually in the Ascomycota family. So there's two that are beneficial, like Penicillium and Saccharomyces, and there are two that are, or a few I should say, that are not beneficial, like Aspergillus, Candida, albicans, and I'll even throw Trichophyton in this family. So the ones that are beneficial, what's Penicillium?
Bread mold.
Yeah, this is technically bread mold, but it's used to make what? Penicillin. The first antibiotic. So that one is obviously helpful. What's Saccharomyces do?
fermentation.
Yeah, this makes fermented beverages like wine and beer. But it's also used to make some types of breads. So this is yeast that's used for that. So these two are obviously beneficial. These three over here are not. Aspergillus is that real pretty aqua green fungus you looked at in lab. But what can it do? It can cause upper respiratory infections. So if a patient inhales a spore, they can get a really nasty upper respiratory infection. Candida albicans is what causes yeast infections. It can also cause thrush. And then trichophyton is the #1 fungus that causes what?
Ringworm.
Ringworm. Skin infection that looks like a red worm, but it's not a worm, it's technically a fungus infection. And it's fairly common, okay? So these are the ones I want you to know. A couple of them are beneficial, Penicillium, Saccharomyces. 3 of them are not, Aspergillus, Candida albicans, and Trichophyton. So definitely know these guys. I think the last part of this chapter is, jump ahead here. Parasites. You have helminths, which are parasitic worms. Those are technically eukaryotes. And then you have the biological vectors, the mechanical vectors. So once again, these are insects or arachnids that either bite you and spread a pathogen, or mechanical vectors, they have pathogens on their body and they walk across your food. So of the biological vectors, the ones that actually bite you, the worst would of course be the mosquito. The mosquito can transmit hundreds of the diseases. And then the ticks are probably #2 that can transmit, Lyme disease, Rocky Mountain spot fever, a few other things. And then historically the flea, which can transmit bubonic plague. So those are the main ones. And with that, that is the end of chapter 12. Now we're going to hit the highlights of chapter 13. Big thing with chapter 13, it's viruses, viroids, and prions. So you should know the general info about a virus. So you guys tell me, what's a virus?
Acellular.
It's acellular, it's not made of cells, and?
It's a single strand of RNA or DNA.
Surrounded by a protein coat, capsid. And it's an obligatory parasite. So those are 3 big things with viruses. They are acellular. They're one of the very few life forms that are not made of cells. 2, they're just a little bit of nucleic acid, either DNA or RNA, but never both. And they're surrounded by a protein coat called a capsid. And they are obligatory parasites. What do I mean when I say that?
You need a host.
They have to have a host to survive. So they cannot grow without a host. They cannot go through metabolism without a host. They cannot reproduce without a host. They have to have a host to survive. So those are 3 big things. A viron is just the extracellular state of the virus. That's the virus before it's penetrated a host cell. It's got nucleic acid, and then it's got a protein coat surrounding it called a capsid. And once again, nucleic acid in here, capsid surrounding to protect the nucleic acid. And once again, with viruses, they can be DNA virus or an RNA virus, but never both. They have to be one or the other. They're very small, smaller than the smallest bacteria. So obviously you need an electron microscope to see them. There is a lot of diversity with their capsids and their shapes. Smallpox virus looks like a pufferfish. Poliovirus looks like a little diamond. And then of course you got bacteriophage T4, which looks like an insect, but it's not. But there's a wide variety. Some of them are cylinder in shape, others are circular. It just depends. And of course this guy. which I have no words to describe how this looks. It looks like something out of a movie. It's got its nucleic acid up here, here's the capsid, and it's got proteins that make a little body, teeth, and legs. What's an envelope?
It's a disguise.
Yeah, it's a fake plasma membrane. About half the viruses that infect mammals, which would include us, they have what's called an envelope, which is a fake plasma membrane. It's a phospholipid bilayer with some glycoproteins. And it's really a disguise, so your immune system doesn't recognize the virus as a pathogen. The good news is enveloped viruses are actually easier to kill than viruses without an envelope. Bad news is they're a lot harder to detect because of this disguise. But that is what an envelope is. Just think of it as a fake plasma membrane that's a disguise. Talking about specific viruses, once again, you divide the viruses up into the DNA family and the RNA family. Within the DNA family, you have smallpox, you have herpes type 1 and 2, you have the Epstein-Barr virus, which causes mono or mononucleosis. You have HPV, which can cause genital warts and cervical cancer. You also have hepatitis B. So those are some of the more important DNA viruses. As far as RNA viruses, especially single-stranded RNA viruses, you've got polio, hepatitis A, multiple ones that cause gastroenteritis, especially in children, like the norovirus and the astrovirus. You've got measles, you've got mumps, you've got rubella, which is German measles. You've got coronavirus, which causes the cold. It also can cause COVID-19. You have Hep C, you got yellow fever, you got influenza, which causes the flu, you got HIV, which causes AIDS. So you have a whole wide variety. You also have rabies, Ebola, Marburg, among others. So the majority of the viruses that affect humans are of what family?
RNA,
Single-stranded RNA. Be aware of that. The majority of viruses that affect humans are of the single-stranded RNA variety. Now, as far as viral replication, most of them go through what's called lytic replication. We have attachment, entry, synthesis, assembly, and release. Attachment is where the virus basically hooks up with the host cell. In this case, we're showing bacteriophage T4. By the way, what's a bacteriophage?
Infects bacteria.
Yeah, it's just a virus that attacks bacteria. First, it's going to attach, and then the entry phase is going to inject its nucleic acid into the bacteria. Then the synthesis is that nucleic acid is going to take over the bacteria and have it only make viral proteins. Then the assembly stage, the bacteria is going to take those viral proteins and make a whole bunch of baby viruses. And then finally you get the release stage where all those baby viruses burst out of the bacteria cell, killing the host cell, and then go on to try to infect other cells. So this is the lytic replication cycle that's been shown with bacteriophages, but this is thought to be how the majority of viruses actually work. There is a variation of this called lysogenic replication, and it's just basically where the virus basically overtakes the cell but lays dormant for a few generations and then decides to go into synthesis and assembly and release all at once. So that's the only difference with lysogenic replication. Three ways that animal viruses can penetrate a cell. One is direct penetration, where the virus does not have an envelope, so just lays on top of the plasma membrane and then injects its nucleic acid into the host cell. The other is membrane fusion. We have an envelope virus. It kind of lays on top of the plasma membrane of the host cell, then just kind of merges and injects its capsid, and then the capsid kind of cracks open and releases the nucleic acid. And then you also have endocytosis, where the cell actually eats the virus by mistake. And once inside the cell, it's got to get rid of its envelope. It then gets rid of its capsid, and then the viral nucleic acid is now in the cytoplasm. Now, the big difference between DNA viruses and RNA viruses, DNA viruses do what?
Attack the nucleus.
They go right to the nucleus of the cell. So DNA viruses go to the nucleus of the cell, and they basically take over the nucleus and have the nucleus only produce viral messenger RNA. RNA viruses are a little sneakier. They do their dirty work in the cytoplasm of the cell. And they do it by one or two methods. They either hijack the ribosomes and have the ribosomes only make viral messenger RNA, or they just trick the ribosomes. They fool the ribosomes in thinking they are messenger RNA, but the end result is the same. The ribosomes only produce viral proteins instead of proteins that are needed by the cell. So just remember, DNA viruses go for the nucleus, take over the nucleus. RNA viruses stay in the cytoplasm and take over the ribosomes. But the end result is the same. You're only making things to make viral proteins. Okay. Let's see what else is important. About 25% of cancers have been related to viruses. Do you guys remember what viruses do?
Image.
Because more
Proto-oncogenes. Proto-oncogenes are like your brakes, if you will, within the cell. They tell the cell when it's time to stop and not go through mitosis. So think of the proto-oncogene as kind of the safeguard. So your cells don't go through mitosis when they don't need to. If you destroy those proto-oncogenes, it's like cutting the brake lines to your car. There's no brakes. So now that cell is going to go through mitosis, until it develops into a tumor, and then cancer, and then it spreads. Well, to get rid of a proto-oncogene, usually you need a couple of insults. And things that can damage proto-oncogenes are too much UV light exposure, radiation, carcinogens like benzene in the water, tar and tobacco smoke, or viruses. Viruses are something that can affect these proto-oncogenes. So cancers that have been known to be caused by viruses would be Burkitt's lymphoma, Hodgkin's lymphoma, Carpi sarcoma, which you see with AIDS patients, or cervical cancer caused by HPV. So about 25% of cancers are thought to be related to viruses. Can you grow viruses in Petri dish?
No.
No, what do you need?
So,
Well, you need a cell. So you can use a cell culture, but honestly, most of the time they use chicken eggs. Because a chicken egg, quite frankly, is just a giant cell. It's very cheap and easy to get, and it's very good at developing viruses. Just remember, a virus must have a host cell to survive. If it doesn't have a host cell, it does not survive. Okay. Now we're going to talk about viroids. What's a viroid? It does. Is it a virus?
It's a cousin.
No. It's similar. It's like a cousin of the virus. It's a small circular piece of single-strand RNA, but it does not have a capsid. So there's no capsid. That's why you can't call it a virus. And it tends to infect plants. And this could be serious because it can pretty much wipe out crops and things like that. But that's the big thing with viroids, is they tend to infect plants. Now, you guys tell me, what's a prion? It's one of our all-time favorites.
Mutated infectious
It's
protein.
a mutated infectious protein. Remember, this is also a cellular pathogen. It's a mutated infectious protein. What condition does it cause?
Mad cow.
Well,
Spongiform
Spongiform encephalopathy. Remember that. Prions cause spongiform encephalopathy, or spongy brain. Encephalopathy just means infection of the brain. Spongiform means the brain's got a bunch of holes in it like a sponge. This is 100% fatal. So what's the big deal with the prion? Well, the big deal with the prion is it resembles a natural protein we have in the plasma membrane of the neuron. Within the plasma membrane of the body of the neuron, there's a protein called PRP. The prion is like a mutated form of PRP, where normal PRP is made up of alpha helix formations at the secondary structure. The prion is made of beta sheets. Well, what's the big deal with that? Well, the big deal with that is if that prion comes in contact with your natural PRP proteins, what does it do? It basically converts your natural PRP into the prion formation. So each time the prion comes in contact with one of your PRP proteins, it causes that PRP protein to switch from alpha helix to beta sheets. So basically these proteins within the plasma membrane of the neurons are changing shapes. Is that a good thing? No, that's a bad thing. Because this plasma membrane was designed to have proteins shaped like this. It was not designed to have proteins shaped like that. So every time you change the shape of the protein, it's like poking a hole in the plasma membrane. Water and fluids are going to rush in, and eventually the neuron dies from osmotic forces. So that's why prions are no good. Prion diseases you should know. In animals, you have bovine spongiform encephalopathy, better known as mad cow disease. In deer, you have chronic wasting disease. And in sheep, you have scrapie. The ones to know in humans are Creutzfeldt-Jakob disease, which is usually abbreviated CJD, and Kuru. Kuru is the one that the tribe in Papua New Guinea got because they were eating brains of dead relatives, which is not good. Another big problem with prion is as of now, there's no cure. And normal sterilization procedures don't kill prions. The only thing that kills prions is cremation. That's why I was mentioning the other day, if there's a prion disease, the whole hospital shuts down, the CDC gets involved, they send dudes in with hazmat suits. Anything that patient remotely came in contact gets carried away and gets cremated. Because cremation is the only thing we found that kills prions. On a positive side, Japan's actually working on a prion vaccine. They haven't finished it yet, but they're working on it. Europe is trying to make an enzyme that will kill prions. So if you caught somebody early enough with CJD, you give them an IV infusion of these enzymes that would hunt down and kill the prions. But those are still both experimental. They have not finished working on those yet. So there is hope for the future, but as of now, prions are 100% fatal. Okay. And that is chapter 13. Any questions? All right, very briefly, your lab tests. Going to be really straightforward. You need to know the gram stain. You need to know the acid-fast stain. And you need to know the endospore stain. With these, you need to know the purpose. So what's the purpose of the gram stain? Basically distinguishing, gram positive versus gram negative bacteria. Gram positive turns what color?
Purple.
Purple. Gram negative turns pink. You need to know the reagents. So you need to know the steps. That's crystal violet. It's iodine. It's alcohol or ethanol. And then it's safranin. I'm not going to ask you like how many seconds for each. It's a little too nitpicky. But you do need to know the reagents. You do need to know what order they go in. So crystal violet does what? It stains.
Purple.
Yeah, it stains gram positives purple. What's iodine do?
It's a hoarded sealer.
Iodine is a mordant. Remember that. A mordant is basically a sealant. Its job is to make sure the crystal violet stays locked in to the gram-positive organisms. So that's the purpose of the iodine, is a mordant. Ethanol is a decolorizer. So what's the big deal with that?
It'll take the color.
You're trying to get the excess stain off the glass slide. So if you don't do that, then your whole glass slide is going to look purple. It's not going to do you any good. You only want your microorganisms to be stained. You don't want the whole slide to be stained. So that's why you use ethanol 95%. You only keep it on maybe 10 to 15 seconds. And it's supposed to decolorize the excess stain you don't need. And then what's safranin do?
It's the counter stain.
It's the counter stain.
And it gets the
Yeah, it's the counter stain
negative.
that basically makes the gram negatives pink. Okay? So that's the main thing you should know. The other thing you should know is what some of the common organisms we've looked at look like with a gram stain. So what does staph look like? We'll say Staph aureus look like with a gram stain.
Purple clusters. Purple cocci.
Yeah, you'd have gram positive cocci in clusters. What would strep look like? It'd be gram positive cocci
And change.
in chains. And what would say like E. coli look like?
Pink.
Just pink rods. Okay? So you should be familiar what common microorganisms look like with a gram stain. Staph would be gram positive cocci in clusters. Strep would be gram positive cocci in chains. E. coli would be pink rods. All right, that's your gram stain. Acid-fast stain. Why do we use acid-fast stain? To look at Mycobacterium bacteria. So that'd be the bacteria that makes tuberculosis, also the bacteria that makes leprosy. And you're basically staining for a mycolic acid, which is a type of lipid that makes up the cell wall. So what are the reagents with this one? Well, you've got quinine, also known as carbonyl fusion, but we'll just say quinine because that's the more common name. You put that on. Then you use a little bit of alcohol as a decolorizer. Sometimes you can use acid alcohol, but I prefer regular alcohol because my experience, acid alcohol would just wipe everything out. And then what's the third reagent?
Methylene blue.
Methylene blue. Blue. And that's the counter stain. So the quinine will actually stain Mycobacterium, and they'll come out as kind of purple or pink rods. Methylene blue is the counter stain. Basically, everything else is going to come out blue. Any other microorganisms you may have are blue. All right, so that's the big stuff with the acid fast. The endospore stain, which we watched a video on, the video is on Blackboard Ultra if you didn't see it. If you go to the lab portion of the course and scroll on down, there's a segment with videos, and they have a video of the endospore stain. But what are basically the ingredients? Well, the main one is malachite green. Malachite green is what actually stains the endospores. Then you can use some alcohol as a decolorizer. Then you'll actually use safranin as a counter stain for the bacteria. So what you'll see is you'll see bacteria that stain pink, but the actual endospores will be green. So you'll see the bright green endospores within like pink bacteria. So that's the whole purpose of the endospore stain. And remember, what two family of bacteria make endospores? Yeah, Clostridium family and the Bacillus family. Okay. Review the handouts I gave you, but this is the meat and the potatoes of the lab test. I can't promise this every single question I could ask, but it's a big portion. You may also be asked to look at slides to identify stuff that we've seen before, like aspergillus or penicillin or helminth, but I will give you a multiple choice, basically selection. So you don't have to remember how to spell or anything. It's just like, what is this? It's aspergillus or it's penicillin or something like that. So you may have 4 or 5 slides to look at. Any questions? That's pretty much it, guys.